Ladies and gentlemen, welcome to a discussion on one of the most exciting yet controversial advancements in modern science, CRISPR. Imagine a world where genetic diseases can be cured, crops can be engineered to withstand harsh climates, and even the extinction of endangered species can be prevented. This is the revolutionary potential that CRISPR technology promises. But like any powerful tool, CRISPR also comes with its own set of risks and challenges that we must carefully consider. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, has taken the scientific world by storm. It's a groundbreaking technology that allows scientists to edit genes with unprecedented precision.
CRISPR is a powerful gene editing tool that allows scientists to make precise changes to the DNA of living organisms. It works like molecular scissors, cutting DNA at specific locations so that genes can be added, removed, or altered. This technology is based on a natural defense mechanism found in bacteria, which they use to fend off viruses. Scientists have harnessed this mechanism to target and modify genes in a wide range of organisms, including humans, plants, and animals. In medicine, CRISPR holds the promise of curing genetic diseases such as cystic fibrosis, sickle cell anemia, and muscular dystrophy. By correcting the faulty genes responsible for these conditions, CRISPR could offer permanent cures rather than just managing symptoms. Additionally, CRISPR is being explored for its potential to combat cancer by targeting and destroying cancerous cells. In agriculture, CRISPR can be used to develop crops that are more resistant to pests, diseases, and environmental stresses.
One of the most significant challenges associated with CRISPR technology is the risk of off-target effects. Off-target effects occur when CRISPR unintentionally edits DNA at locations other than the intended target. This can lead to unintended genetic changes, which may have unpredictable and potentially harmful consequences. Understanding and mitigating these off-target effects is crucial for the safe and effective use of CRISPR. Research has shown that off-target effects can occur with varying frequencies depending on the specific CRISPR system and the organism being edited. Scientists are actively working to understand the mechanisms behind off-target effects and develop strategies to minimize them. Despite these efforts, the risk of off-target effects cannot be entirely eliminated, highlighting the need for rigorous testing and validation before CRISPR applications can be widely adopted.
Efforts to minimize off-target effects are at the forefront of CRISPR research. Scientists are employing various strategies to enhance the precision and safety of this powerful gene editing tool. One approach involves the development of improved CRISPR systems that are less prone to off-target effects. For instance, researchers have engineered versions of the CRISPR-Cas9 system that have higher specificity and reduced off-target activity. Another promising strategy is the use of computational tools to predict potential off-target sites before conducting experiments. These tools analyze the genome to identify regions that share similarities with the target site and are therefore at a risk of being inadvertently edited. Additionally, researchers are exploring the use of alternative CRISPR systems such as CRISPR-Cas12 and CRISPR-Cas13, which may offer different advantages in terms of specificity and efficiency. The continuous development and refinement of CRISPR technologies are essential for achieving safer and more reliable gene editing.
Section 5. The unknown long-term effects, a call for caution. While CRISPR technology holds immense promise, the long-term effects of gene editing remain largely unknown. This uncertainty is a significant challenge. The potential for unintended consequences highlights the need for a cautious approach. Genetic changes could have unforeseen effects on health. A gene edit could disrupt other important genes, leading to new health problems.
Section 6. Speculating long-term risks, unintended genetic changes and cancer. CRISPR's potential long-term risks include unintended genetic changes leading to cancer. Cancer arises from mutations, and gene editing could increase this risk. Understanding CRISPR's full effects is crucial. CRISPR mutations might disrupt tumor suppressor genes or activate oncogenes. Thorough investigation into CRISPR's carcinogenic effects is needed. Scientists are exploring strategies to mitigate these risks.
Section 7. Ethical challenges and societal implications of human gene editing. The advent of CRISPR technology has brought with it a host of ethical challenges and societal implications. Human gene editing raises profound ethical questions about our genetic makeup and the impact on future generations. Addressing these challenges is crucial for the responsible use of CRISPR. One primary concern is the potential for designer babies, enhancing traits beyond health reasons. This raises questions about fairness and genetic discrimination. Ensuring ethical considerations in CRISPR research is essential.
Section 8. The regulatory landscape toward robust oversight. The rapid development of CRISPR technology has outpaced the establishment of comprehensive regulatory frameworks to govern its use. Ensuring robust oversight is essential to address the potential risks and ethical challenges associated with gene editing. Current regulatory approaches vary widely across different countries, reflecting diverse perspectives on the balance between innovation and precaution. In the United States, the Food and Drug Administration, or FDA, and the National Institutes of Health, or NIH, play key roles in regulating gene editing research and applications. The FDA oversees the safety and efficacy of gene editing therapies, while the NIH provides guidelines for ethical research practices. However, the regulatory landscape is still evolving, and there is a need for clearer guidelines and more coordinated oversight to address the unique challenges posed by CRISPR. The need for robust oversight extends beyond medical applications to include agricultural and environmental uses of CRISPR.
Section 9. Gene drives and ecological risks. Impacts on biodiversity. Gene drives are a powerful application of CRISPR technology that can spread genetic changes rapidly through populations. This is achieved by biasing the inheritance of a particular gene, ensuring that it is passed on to a large proportion of offspring. While gene drives hold great promise for addressing issues such as disease control and pest management, they also pose significant ecological risks and potential impacts on biodiversity. One of the primary concerns with gene drives is their potential to cause unintended ecological consequences. For example, releasing a gene drive to control a pest species could disrupt entire ecosystems by affecting the species that prey on or compete with the targeted pest. This could lead to cascading effects that harm biodiversity and ecosystem stability. The long-term ecological impacts of gene drives are difficult to predict, making cautious and measured use essential.
Section 10. Public engagement and addressing misconceptions. Public engagement is a critical component of the responsible development and use of CRISPR technology. Ensuring that the public is informed about the potential benefits and risks of gene editing is essential for building trust and fostering informed decision-making. Addressing misconceptions and providing accurate information can help to demystify CRISPR and promote a balanced understanding of its implications. One common misconception is that CRISPR technology can provide simple and immediate solutions to complex problems. While CRISPR holds great promise, it is not a panacea. Gene editing involves complex biological processes, and the outcomes are not always predictable. Communicating the uncertainties and limitations of CRISPR is crucial to manage public expectations and avoid unrealistic hype. Another important aspect of public engagement is involving diverse voices in the discussion about CRISPR. Section 11. Summary and call to action, responsible use, and continued research. In this essay, we have explored the revolutionary potential of CRISPR technology and the various risks and challenges associated with its use. From unintended genetic changes and long-term effects to ethical dilemmas and regulatory issues, the responsible development and application of CRISPR require careful consideration and robust oversight. CRISPR holds immense promise for transforming medicine, agriculture, and environmental conservation. However, the potential for off-target effects and unknown long-term consequences underscores the need for rigorous research and validation. Efforts to minimize these risks through improved CRISPR systems and advanced computational tools are crucial for ensuring the safety and efficacy of gene editing applications. Section 12. Conclusion, invitation for interaction and gratitude. Thank you for joining us on this exploration of the risks and challenges associated with CRISPR technology. We hope that this discussion has provided valuable insights into the complex